Hello everyone, welcome back to our channel Arunodayam Biology. Today we are going to learn about an activity to prove that chlorophyll is essential for photosynthesis from life processes chapter. Chlorophyll is essential for photosynthesis. Already we know that plants need four raw materials to perform a process called photosynthesis. Those four raw materials are chlorophyll, sunlight, water and carbon dioxide. So now we are going to learn how chlorophyll is essential for photosynthesis. So for that, first of all, we have to take a leaf and take cross section of it. Take a cross section of leaf and place it under microscope. Observe it. If we observe a cross section of leaf under microscope, we can see many green colored dot like structures in it. So already we know that every living organism's section has a cells in it. So of course, if we take cross section of leaf, we can find so many cells. In those cells, we can notice green colored dot like structures. These green colored dot like structures are nothing but a cell organelles called chloroplasts. Cell organelles called chloroplasts, and each chloroplast contain a green color pigment called chlorophyll. And this chlorophyll is useful for observing a certain wavelength of light from sun, and thus helps in the process of photosynthesis. Already we have learnt it in our previous video. So it is clear that every leaf of a plant has some green dot like structures called as chloroplasts, and these chloroplasts has a green color pigment called as a chlorophyll. Now we have to show that this chlorophyll is essential for photosynthesis. For that we have to do one activity. For this activity we have to take a potted plant with variated leaves. Variated leaves means a leaf which contains colorless areas and green color patches. A leaf which has both colorless areas and some green color patches, those type of leaves are called as variated leaves. We have to take any potted plant with variated leaves. For example, money plants or croton plants, we can find these variated leaves on money plants or croton plants. We can take any of this potted plant. Next, we have to put this potted plant in dark room for 3 days. We have to put it in dark room for 3 days. Why? The starch which is present in the plant will have to get used up. If we put a potted plant in dark room, it cannot perform photosynthesis, isn't it? Why? Because sunlight is not available to it. So without photosynthesis, how will the plant survive? It will not get food, no? So within these 3 days, the plant, the leaf and the plants will use the already stored starch present in their body, just like us. So we also store some fatty substances under our skin in the adipose tissue. We all know about it, isn't it? We will store some energy in the form of fat under our skin. If we are under starvation, if we do not take any food material for 3 or 4 days, we will use the fat which is stored under our skin for energy, isn't it? So just like that, in case of plants also, they will store energy or food material in the form of a starch. They will store a food material in the form of a starch. So when we put this potted plant in dark for 3 days, as the plant cannot perform photosynthesis, and in order to meet its daily energy, the plants will use the stored starch present in their leaves. So within these 3 days, the starch will get used up. Next, now keep the potted plant in sunlight for about 6 hours. Now take the potted plant from the dark room and put it in sunlight for 6 hours. So during these 6 hours, the plant will perform photosynthesis. Okay. Next, after that, pluck a leaf from the potted plant. Choose any leaf and pluck it. It has both green color areas and colorless areas, already I said. So take a white color paper and trace the green color areas. So mark these green color areas with a marker. Take a white paper and trace out these white color patches on it. Okay, we have to use it at the end of the experiment. You have to use the white sheet at the end of the experiment. Now clear now? Now dip the leaf in boiling water for few minutes. For few minutes we have to put this leaf in boiling water. Then after, we have to dip or put this leaf in a beaker containing alcohol. We have to put this leaf in a beaker containing alcohol and place the beaker with alcohol and leaf into a water bath. Do you understand? We have to put this leaf in a beaker containing alcohol and we have to transfer that beaker along with alcohol and leaf into a water bath. We have to boil it for few minutes. Why we are doing like this? Why? Because we have to boil the leaf in alcohol but not directly on the flame. Why? Because alcohol easily catches fire. So in order to avoid fire accidents, we are putting this leaf in alcohol in a beaker and then we are boiling it on a water bath, not directly on the fire. Okay, then after some time we can see the color of the leaf. After boiling the leaf in alcohol for some time in water bath, we can clearly see that the leaf of the color changes. We can see that the leaf completely becomes colorless. Leaf completely becomes colorless. Why? Because the green color areas also becomes faded. The leaf becomes totally colorless. Now dip the leaf in a dilute solution of iodine for few minutes. And now take off the leaf from the alcohol and dip it in a dilute solution of iodine for few minutes. Iodine, it is a reagent which is used to test the presence of a starch in the leaves. Okay, we have to put this leaf in a watch glass containing dilute solution of iodine for few minutes. Then after, we have to wash the leaf. We can clearly see that some areas of leaf turns into blue black color. Some areas of leaf turns into blue black color. That too, those areas which are green before the test. When we compare this leaf with the white sheet which we have traced the green patches in the starting of the experiment, we can clearly see that the areas which have green color earlier only turned into blue black color. The areas which are colorless remain colorless even after the experiment also. Why? Because these green color areas or these green color patches contain actually chloroplasts. In turn, they contain chlorophyll pigment. So these chlorophyll pigments absorb sunlight and performed photosynthesis only in these areas. And they prepared food material. Okay. So after iodine test, only these areas are turning into blue black color, indicating that this areas contains a starch. Why? Because only chlorophyll is present here, and the areas which were colorless remain colorless only. Why? Because these areas do not contain chlorophyll. That's why they do not perform photosynthesis. That's why they do not contain food material also. So that is about our activity today in our video. So we have learnt about an activity on how chloroplast is essential for photosynthesis. So for that we have taken a cross section of a leaf and observed it under microscope. It has so many green dot like structures called chloroplast containing chlorophyll. Now we have started an activity. We took a potted plant with variated leaves and put it in 3 days dark room in order to make the starch used up. After that we have taken that potted plant and put it in sunlight for 6 hours. After 6 hours, we have taken this leaf, we have plucked one of the leaf from the plant and we have traced the green color areas with a white paper. Then after, we dip the leaf in boiling water for few minutes and then into a beaker containing alcohol, and we transferred the beaker into a water bath and boiled it for few minutes. After some time, we have taken this leaf out of the alcohol and rinsed it with a dilute iodine solution. After some time, we can clearly see that only the areas which have chlorophyll is turning into blue black color, showing that they have starch, and the colorless areas are showing that they do not contain chlorophyll. That means they do not perform photosynthesis. So that is about our video today. If you have any doubts, please ask me in comment section. I will clarify you. Hope you understand this video. Thank you so much for watching. Jai Hind.